In 3, 2, 1, go.
Good day, BS Biology 3B and Ma'am Arieta. We are the Group 4 to present to you our journal entitled The Relevance of Knocking Out GGTA1 Genes in Porcine Cells for a Successful Xenotransplantation.
Human organ transplantation is a growing field of medicine which is now constantly faced with a significant challenge. The scarcity of deceased human organs and cells for clinical transplantation remains a major concern around the world. This issue might be overcome by transplanting organs from pigs that have been genetically modified to resist the human immune response. Since according to Butter et al., Cooper et al., and Rai Horowitz et al., pigs are genetically, anatomically, and physiologically similar to humans, making them the ideal donor of biological materials for xenotransplantation. The huge discrepancy between the number of patients waiting to receive transplants and the available donor organs and tissues, including hearts, livers, kidneys, lungs, islets, and corneas, suggests that xenotransplantation using pig tissues or organs has been suggested as a promising solution to the donor tissue or organ shortage. However, there is a substantial phylogenetic distance between pigs and humans, which can cause immune system problems following xenotransplantation, stated by Ray Horowitz et al. 2017. It is critical to generate and characterize transgenic pigs with reduced immunogenic properties in order to facilitate successful xenotransplantation.
The following classification were used based on the mechanism and timing of xenotransplant rejection. Number 1 is the hyperacute. Hyperacute rejection occurs within several minutes of organ transplantation that leads to failure. This rejection results in endothelial deteriorations and loss of its biological characteristics. The capillaries and arterioles undergo pathological alterations. The main reason for the hyperacute rejection is the presence of natural antibodies in human plasma that detect the gal alpha 1,3 gal antigen on the surface of porcine endothelial cells. The recognition and binding of the gal alpha 1,3 gal antigen by xenoreactive antibodies activates the classical complement pathway, resulting in the formation of the membrane attack complex, which acts as a catalyst for cell membrane penetration by proteins, forming transmembrane channels, eventually leading to cell lysis. The number 2 is delayed xenograft rejection, is a xenogenic transplant which avoids hyperacute rejection, is subjected to delayed xenograft rejection taking place several hours to several days after organ transplantation. The course of delayed rejection resembles closely hyperacute rejection, with this difference that it does not require the participation of the complement. It involves IgG immunoglobulins, and it develops more slowly and is initiated in the arterial lumen and not in capillaries and arterioles. The acute chronic rejection occurs within several days after transplantation. The dominant morphological characteristic of such rejection are infiltration with predominant mononuclear cells located interstitially in tissues. In the human, the cellular response targeting porcine tissues is induced by the NK cells and the T lymphocytes, with the CD4 plus lymphocytes exhibit a much higher level of cytotoxicity than CD8 plus lymphocyte. And lastly, the chronic rejection. It occurs within months or even years after transplantation, and it is clinically manifested by progressing failure of the grafted organ or tissue. It is assumed that chronic rejection develops as a result of additive effects or various harmful factors. At present, little is known on chronic xenotransplant rejection. It is suggested that the initiating factor may be connected with damage of vascular endothelium by cytotoxic T lymphocytes and specific antibodies. However, the intensity of the response is much lower than in the case of acute rejection. In cell membranes, we observe increased expression of cell adhesion molecules and the tissue factor activating the coagulation system. Gradual proliferation of the aortic media takes place, leading as consequence to vascular obliteration and organ failure.
I'm Joshua Prince de la Cruz of group number 4. I will be discussing the statement of the problem. The main thrust of this study is to evaluate the importance regarding the deletion of the enzyme GGTA1 in porcine cells as a key factor for a successful organ xenotransplantation between pigs and non-human primates, wherein their cells may also be used as a basis of pig to human xenotransplantation models. Moreover, knocking out the GGTA1 enzyme will be a big factor in order for the organ to overcome the possibility of being rejected by the human body during xenotransplantation. Specifically, the researchers seek to answer the following questions. First question, how does knocking out GGTA1 overcome xenograft rejection? Second question, what is the maximum lifespan of the organs genetically modified? Third question, why are the pigs the ideal donor of xenotransplantation? Fourth question, what are the possible risks in xenotransplantation? And lastly, based on the study, how would genetically modified pigs affect the field of medicine?
I am Patricia Nicodemus and I am going to discuss the research design. Our group ought to prove the hypothesis that knocking out GGTA1 alpha 1 transferase genes in porcine cells have a relative impact to the success rate of xenotransplantation. Therefore, we utilize a correlational research design. It is a non-experimental research of predicting and explaining the link between variables. Furthermore, correlational research findings can be used to assess free valence and relationships between variables, as well as predict occurrences based on current data and knowledge. The researchers decided to make use of this research design since the study aims to explain whether there's a positive correlation between the main variables, knocking out GGTA1 genes in porcine cells, and successful xenotransplantation. A positive correlation exists when two variables move in the same direction or are directly proportional with each other. In other words, it is when one variable decreases as the other decreases or when one variable increases as the other increases. For instance, it can be observed when a successful GGTA1 knockout results in a successful xenotransplantation. In order to present and analyze the data that were collected in this study, we use a method of correlation and regression, specifically the Spearman rank correlation. This non-parametric correlation analysis is used to determine the degree to which two variables are related. It is it necessitates sorting the data and assigning a specific rank to each value, with 1 being the lowest. Furthermore, because Spearman assesses the strength of a monotonic association, the data must be related to each other in a monotonic manner. This basically means that if one variable rises or falls, the other rises as well or decreases. The formula for this analysis or correlation coefficient is P equals 1 minus 6 summation of D1 raised to 2 over n parentheses n squared minus 1. P means Spearman's rank correlation coefficient. D is the difference between the two ranks of each observation, and N is the number of observations. Where the result P equals 1 denotes a perfect positive correlation, and P equals negative 1 denotes a perfect negative correlation. The researchers performed the analysis by first identifying the variables that they will use in the computation that may represent the main variables. They make use of the gathered data regarding the life expectancies of wild-type WT and genetically modified GDKO pigs. A table was then made and ranked them, being the longest life expectancy ranked 1. The difference between the paired ranks was given and squared. Lastly, the computation was made by plotting the data in the Spearman formula.
To establish the relationship between xenotransplantation and the deletion of enzyme GGTA1 in porcine cells, the researchers gathered different existing studies consisting of compiled analysis of data that is composed of graphs, models, and tables. So for the procedure, The researchers ensured that the data that they acquired were information rich and reliable. Afterwards, they evaluated and analyzed the results and conclusions of several studies that illustrate how the deletion of gene GGTA1 in porcine cells, as well as graphs, tables, and models discussing the prospect of a successful pig-to-human xenotransplantation using non-human primate preclinical data.
Good morning, everyone. I am Patrick Gonzalez, and I will present the results of the study. For the results of the study, the researchers determined that there is a significance in removing gene GTTA1 in porcine cells. This table right here from the study of Liu et al. 2020 shows how long different organs from wild type and genetically modified pigs are able to survive in non-human primates after transplantation. Furthermore, the table also suggests there is a significant difference between the two sets of data, wherein wild type organs are only able to last for a few hours to days, while genetically modified organs last for weeks to years. It is also because of this observation that the researchers use this table as a reference for the following correlational analysis. And the correlation analysis is in our figure 1. With the use of Spearman's rank order correlation, the measurement regarding the strength and direction of association that exists between the maximum life expectancy of different organs from different origins can be obtained. The correlational value between the two variables is negative 2 upon evaluating evaluating. As a result, this value states that there is a negative correlation between wild type and genetically modified organs, wherein it suggests that there is a desirable and undesirable choice between this type of organs. And for that reason, there is based on their maximum life expectancy, the analysis also suggests that the organs with lack of GGTA1 should be utilized to have a more positive impact in xenotransplantation. This can be further explained by the following figures. In figure 2, in figure 2, immunoglobulins regulate the complement system by activating complement on foreign surfaces and diverting reactive complement proteins away from autologous cell surfaces. In other words, they act as a critical part of the immune response, specifically recognizing and binding to particular antigens such as bacteria and viruses and aiding in their destruction. Based on this figure by Yamamoto et al. 2020, there is an observable amount of decrease in immunoglobulin G and immunoglobulin M binding among all of the respondents given GTKO organs when compared to those given with wild type organs. This is due to the fact that the PBMCs of GTKO pigs did not express the gall antigen. Therefore, this figure indicates that the success rate as well as the survivability of xenograft organs can be further increased when gene GGTA1 is removed. Apart from that, from figure 3, cells treating cells with the cytotoxic compound can result in a variety of cell fates. In the case of xenotransplantation, cytotoxicity pertains to the recognition of porcine alpha gal epitopes by the immunoglobulin G and immunoglobulin M and the binding that occurs between these molecules. It is based on the number of cell death found within the sample. As such, it also describes the survivability rate of xenograft organs when introduced to the recipient's body. The figure presents how serums containing the PBMCs of different primate species react with the PBMCs of wild type and GTK porcine cells. If a sample is found to have a high cytotoxicity percentage, hyperacute rejection would occur. However, if a sample is found to have a low cytotoxicity, there's a higher chance of success for xenotransplantation. Based on the data, there is also a slight change or decrease in the percentage between wild type cells and GTKO cells. This indicates that there is an observable effect of knocking GGTA1 to overcoming hyperacute rejection. Now let us move on to the discussion of the study. In xenotransplantation, an aggressive defensive mechanism known as hyperacute rejection takes place, wherein the recipient's immune system sets out to destroy the transplanted organ. This rapid, this rapid destruction of the graft is due to the presence of pre-existing antibodies in humans and non-human primates that bind to porcine specific glycan epitopes heavily expressed on the pig vascular endothelium and activate antibody-dependent cell-mediated cytotoxicity or complement-dependent cytotoxicity mechanism. The main xenoantigen for this event is alpha-gal, synthesized by GGTA1. The findings of this study heavily suggest that the reproduction of genetically modified pigs that should be used for xenoplantation should at least have GGTA1 gene inactivated in an effort to prevent alpha-gal from being expressed in the cell surfaces of the pig. As a result, the researchers believe that this lack of gene will prevent immediate rejection of the graft and waste valuable resources. This assumption can be further supported by the study conducted by Yamamoto et al. 2020, wherein they examined the number of antigen antibody bindings and cell death percentages in different primate species when given blood samples from wild type and GTKO pigs, as seen in figures 2 and 3 once again. Going back to these figures, their study results in New World monkeys, capuchin monkey, and squirrel monkey having an average of relative guidance molecules 4.7 and immunoglobulin M antibody binding to wild type cells, 21.5 in Old World monkey, baboon, rhesus monkey, cynomolgus monkey, and African green monkey, and 31.8 in humans. While in GTKO cells, they have 6.8, 6.8, 6.6, and 19.5 respectively. On the other hand, New World monkeys have an average of 1.6 relative guidance molecules, immunoglobulin G antibody binding to wild type cells, 3.9 in Old World monkey, and 9.3 in humans. For GTKO cells, they have 2.0, 1.2, and 5.7 respectively. As for the cell's cytotoxicity at 25% serum concentration, New World monkeys have a 55.6 percentage, Old World monkeys have 93.6%, and humans have 66.7%. In summary, when one or more pig xenoantigens are deleted, there is a significant reduction in the binding of human antibodies and CDC to pig PBMCs.
Since different organs are used in xenotransplantation, the length of survival also varies. This is because survival time mainly depends on the types of transplanted organs. But with the continuous development of genetic engineering, additional alterations and immunosuppressive techniques, improvements are seen with the organ survival of preclinical pig to non-human primate xenotransplantation models. On the question on why pigs are the ideal donor for xenotransplantation is mainly because it can be genetically modified to overcome rejection mechanisms and physiological incompatibilities, as well as to reduce the risk of spreading zoonotic infections. Discussion on its possible risks in xenotransplantation involves the possibility of transmitting swine microbes such as bacteria, fungus, and viruses that can adapt in the recipient and cause disease. Concerns regarding the risk for endogenous retroviruses are also present. Retroviruses can be activated by particular circumstances within the host and become infectious. Other than that, pigs that have been genetically modified hold great promise in the field of medicine. Their use in xenotransplantation can potentially address the growing problem of organ scarcity. Also, it is cost effective since treatment and intensive care treatment for patients with end stage organ diseases will be less necessary and they can be less reliant on immunosuppressive drugs. On this slide, this table only shows the survival time of the following and different organs from previous studies, and this is also the one that I have discussed earlier.
I'm Rosemary Macario together with Hope Cruz and Faye Suryo to present to you our conclusion and recommendation. The purpose of this study is to find whether the removal of GGTA1 will be beneficial to the success of xenotransplantation. The researcher explore and identify certain aspects like antibodies and porcine cells and how they function in the instant of xenotransplantation. The researcher conclude that the researcher determine and focus on how the removal of GGTA1 can majorly affect the survivability of a patient that underwent during and after xenotransplantation. The survivability rate of a non-human recipient is entirely dependent on whether the organ transplanted was genetically altered or not, the former being the more significant.
Multiple studies correlates the success of xenotransplantation with the genetic background of the donor. The researchers were able to draw multiple assumptions, one being the survivability rate of the recipient was significantly higher when the organ transplanted was genetically modified. This study will help to make students and researchers alike become aware about the possibilities of xenotransplantation and to solve one of the major problems of, of the field of medicine today. And now, to present our recommendations, here is Face to You.
Good day. My name is Faye Suryo and I'm here to present to you our group's recommendations for our journal. We based our recommendations from the findings, conclusions, and review of the study. To start off, the researchers recommended that the following be attained. Number one, the researchers recommend coming up with more complex modulation of the animal to human antibody and cellular recognition. This is to increase the success rate of xenotransplantation and lessen the chances of rejection. Next, the researchers suggest that further improvements could still be achieved by in-depth study in order to fill in the gaps of the research. Then, it is found out that CRISPR-Cas9 has a relatively high frequency of off-target effect. Researchers suggest that a lot of emphasis should be placed on the possibility of reducing the risk of so-called off-targets by enhancing or improving Cas9 protein or sgRNA modifications. To counteract the human immune response to pig organs, it is recommended to establish and obtain an appropriate combination of porcine genome multi-modification to ensure virological safety. Next would be the researchers suggest that the identification of the ideal combination of genetic modifications on the organ source pig is a crucial step to move forward into first in man clinical studies. It is also recommended to use other approaches to knock out genes aside from CRISPR-Cas9 to produce genetically modified large animals with unexpected genotype. Then, it is also found out that the depth of the analysis method may have been limited. Researchers suggest more accurate approaches such as whole genome sequencing that can determine the order of the bases in the genome of an organism in one process. Knocking out GGTA1 may reduce the antibody barrier and increase the success rate of xenotransplantation. The researchers recommend performing laboratory testing in humans in further research studies for it to become feasible. And lastly, researchers suggest testing the three methods in gene knockout, such as the GGTA1 knockout, double knockout, or the triple knockout to know which of them is effective in successful xenotransplantation to avoid rejection.
Thank you, and that's all for our research. Thank you for listening.